Okay, dear friends, let's move to our topic of discussion, which is RNA polymerase. The transcription is nothing but the synthesis of RNA from DNA, which is catalyzed by an enzyme called RNA polymerase. We'll discuss today about the prokaryotic RNA polymerases and eukaryotic RNA polymerases. First, let us discuss prokaryotic RNA polymerases. The prokaryotic RNA polymerase, it is only single type of RNA polymerase is present. Only one type of RNA polymerase which can synthesize all the RNAs. That means it can synthesize that single type of the RNA polymerase can synthesize mRNA, rRNA as well as tRNA, and it is a large multi-complex enzyme with a molecular weight of around 465 kilodaltons. It contains 6 subunits, and these 6 subunits are 2 identical alpha, 1 beta subunit, another beta prime subunit, omega subunit, and the sigma subunit. These are the 6 subunits which are present in the RNA polymerase enzyme. And these are nothing but it is a large multi-complex enzyme. Means remember, this is 2 identical alpha subunits. Imagine these are alpha, this is beta, and this is beta prime. There is sigma and a small subunit omega. This is the large multi-complex enzyme. Remember that each subunit is nothing but a polypeptide chain. That means it is made of protein. Enzymes are nothing but protein. As it is made of polypeptide chain, it is encoded by genes. There is a separate gene for every subunit. Let's see what are those subunits and what are the genes encoded. Okay, let's see. These are the subunits: alpha, beta, beta prime, sigma, omega, and these are the genes encoded. Alpha subunit is encoded by RPOA gene, beta subunit is encoded by RPOB gene, and beta prime it is RPOC gene, sigma subunit is encoded by RPOD gene. We don't know the subunit encoded by omega subunit, and these are the molecular weights. And let's see, before this we will see two terms. One is the holo enzyme and core enzyme. Holo enzyme means in the RNA polymerase, if the RNA polymerase contains all the subunits, that means 2 alpha subunits, beta, beta prime, sigma and omega, then it is known as a holo enzyme containing all the subunits. From the holo enzyme, if sigma subunit comes out, then it becomes the core enzyme. Very simple. From all the subunits, if sigma subunit dissociate, it is called as RNA polymerase core enzyme. And let's see this term also. Polymerization rate is number of nucleotides per second. Number of nucleotides per second is called as polymerization rate. The polymerization rate of RNA polymerase in prokaryotes is about 50 nucleotides per second. Listen, sigma subunit, it must be the part of the holo enzyme. Remaining, they are the part of the core enzyme. And each subunit has a specific function. Alpha subunit has the function of binding to the promoter. What is promoter? Promoters are short oligonucleotides which are consensus and which are present on the upstream of DNA, which regulate the binding of the RNA polymerase. The binding of RNA polymerase will depend on the promoter sequences, and these promoters are present on the upstream. What is upstream? Imagine this lower one is the template strand, this is the template strand, and this is the newly synthesized RNA. RNA synthesis is starting from here, so this is the start point, and this is the template strand. Just to the left hand side These sequences are called upstream sequences. The encoded sequences towards the right are called downstream sequences. Upstream sequences are indicated by negative symbol -1, -2, -3, -10, like that. The promoter sequences are present in the upstream, and there are two types of promoter sequences which we will see later, one at -10 and one at -35. And this RNA polymerase A, it binds to the promoters. Thereby, they after binding to the promoter, they will start the process of transcription by bringing a confirmatory change in the RNA polymerase. And then the beta subunit, which is encoded by RPOB gene, it is the main catalytic site and binds to the NTPs. What are NTPs? Nucleotide triphosphates like ATP, GTP, CTP, TTP, UTP, etc. All these nucleotides are present in the vicinity, and this beta subunit, it will bind to it recognizes and binds to the specific nucleotides and catalyzes the formation of phosphodiester bond. A phosphodiester bond is formed between, imagine if this is the first nucleotide, okay, then a phosphodiester bond is formed, and this is the second nucleotide. Okay, so this formation of phosphodiester bond is done by the beta subunit, and beta prime subunit, it binds to the DNA template strand, facilitates the catalysis. Sigma subunit encoded by RPOD gene, and it is the part of the holoenzyme which is required for the recognition of the promoters. Okay, if this is the template strand and recognition of promoters are present on the upstream, I told you. Here the RNA polymerase, it binds to the promoter. This is the RNA polymerase which is binding to the promoter sequences. For the binding of the RNA polymerase to the promoters has to be recognized. For the recognition of the promoters, this sigma subunit is very useful. So sigma subunit, it helps in the recognition of the promoters. And last one is the omega subunit. It is required for the assembly of the RNA polymerase, and it stabilizes the assembled RNA polymerase. Okay, let's again sum up the subunits. Alpha subunit encoded by RPOA gene, and it is two identical alpha subunits are present which is required for the binding of the promoters. Next is beta subunit encoded by RPOB gene, and it is the main catalytic center, and it is inhibited by the rifampicin. Rifampicin is an antibiotic. Rifampicin, it is an antibiotic which inhibits the initiation process of transcription but not elongation and termination. That means it inhibits the RNA polymerase holoenzyme but not the core enzyme. Rifampicin antibiotic, when added, it inhibits the RNA polymerase holoenzyme only but not the core enzyme. Let's move on to the beta prime subunit encoded by RPOC gene, and it binds to zinc ions which is required for the enzyme, and it binds to the template strand. Sigma subunit, it is the part of the holoenzyme encoded by RPOD gene which is required for the recognition of promoters, and there are two types of promoters in prokaryotes, -10 region and another at -35 region. And this sigma subunit, it increases the affinity of the holoenzyme to bind to the promoter sequences. Hence, if it is a DNA strand and this is the RNA polymerase will be moving in searching for the promoter sequences. Once it finds the promoter sequences, the sigma subunit in the RNA polymerase, it recognizes the promoter sequences, thereby increases the affinity of the holoenzyme to bind to the promoters. And the omega subunit, it is the smallest subunit. It facilitates the assembly of RNA polymerase and it stabilizes the RNA polymerase also. That's all about the prokaryotic RNA polymerase. Let's move on to, let's have a brief discussion on the eukaryotic RNA polymerase also. The eukaryotic RNA polymerases, there are three types of RNA polymerases. Three types are there. One is RNA polymerase 1, RNA polymerase 2, as well as RNA polymerase 3. Main types 4 and 5 are also there. And these different types of eukaryotic RNA polymerases were identified using an antibiotic called alpha amanitin. Alpha amanitin is isolated from a poisonous mushroom called Amanita phalloides. This alpha amanitin, it is made of 8 amino acids and it is a cyclic peptide. All these three RNA polymerases or 5 types of RNA polymerases respond differently to the alpha amanitin. That is why they were identified. And this is the selective inhibitor of RNA polymerase 2 as well as 3, but it do not inhibit RNA polymerase 1. That means RNA polymerase 1 is insensitive to the alpha amanitin, whereas RNA polymerase 2 is highly sensitive. RNA polymerase 1 is insensitive. It is not at all responding even though alpha amanitin is present, the RNA is synthesized. But RNA polymerase 2, it is highly sensitive. That means even at the presence of small amount of alpha amanitin, it inhibits the synthesis of mRNA. Whereas RNA polymerase 3 is moderately sensitive, inhibited at concentration of 10 micrograms per ml of alpha amanitin. RNA polymerase 4 is also slightly sensitive. It needs 50 micrograms per ml concentration of alpha amanitin. Okay, these are the sensitivity of RNA polymerases towards alpha amanitin. Okay, let's see what type of RNAs are synthesized from RNA polymerase 1, 2, 3. These eukaryotes have multiple types of nuclear RNA polymerases. Each RNA polymerase is responsible for the synthesis of a particular subset of RNAs. For example, if we see RNA polymerase 1 synthesizes rRNA, and this rRNA in the beginning it is synthesized in the form of 45S rRNA, ribosomal RNA, and later on it breaks and gives rise to 28S rRNA, 18S rRNA, as well as 5.8S rRNA, which forms the major sections of the ribosomes. Remember, these are the rRNAs which are the part of the ribosome. Ribosome is made of ribosomal RNA plus ribosomal proteins. Okay, these ribosomal RNAs, these are eukaryotic rRNAs. They are required for the formation of the ribosome. And the rRNA is synthesized from which RNA polymerase? RNA polymerase 1. The RNA polymerase 2, it synthesizes mRNA precursors and small nuclear RNAs called as snRNAs. Likewise, microRNAs. They are synthesized from which RNA polymerase? RNA polymerase 2. RNA polymerase 3 synthesizes tRNA, 5S rRNA, and small other RNAs which are found both in the nucleus as well as in the cytoplasm. Remember, RNA polymerase 3 synthesizes tRNA and 5S rRNA. 5S rRNA is part of the ribosomes. It is required for the assembly of ribosome, and it also synthesizes tRNA. RNA polymerase 4 and 5, they are found in the plants and they are usually less understood and we don't mention in the RNA polymerases, but they make some siRNA, small interfering RNAs or silencing RNAs which are required for the regulation process. Okay, briefly these are the eukaryotic RNA polymerases. Okay, let's sum up the eukaryotic RNA polymerases. There are three types of RNA polymerases in eukaryotes. They are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and their sensitivity to alpha amanitin is also different. RNA polymerase 1 is insensitive, RNA polymerase 2 is highly sensitive, RNA polymerase 3 is moderately sensitive. RNA polymerase 1 synthesizes rRNA, RNA polymerase 2 synthesizes mRNA as well as snRNA, RNA polymerase 3 synthesizes tRNA, 5S rRNA, and small RNAs in the cytoplasm as well as nucleus. That means even though alpha amanitin is present, we are going to get ribosomal RNA, but mRNA synthesis is highly inhibited by the small amount of the alpha amanitin. At a concentration of 10 micrograms per ml, the tRNA synthesis is also inhibited. Okay, these are the eukaryotic RNA polymerases, RNA polymerase 1, 2, and 3. And the prokaryotic RNA polymerase is only one type of RNA polymerase is present, which synthesizes all the mRNA, tRNA, as well as rRNAs. That's all. Thanks for watching. Do introduce to your friends. If you like this video, please share and comment and subscribe. Thank you. Thanks for watching.